Right, good morning. We're going to keep talking about gene transfer between species. So individuals usually inherit DNA from their parents. Surprisingly, scientists have discovered that genes can also pass between individuals from different species. The transfer of genes can happen when one cell engulfs another, or when bacteria share their DNA with other cells. The transferred DNA is almost always destroyed, but occasionally a cell adds new genes to its DNA. Bacteria use gene transfer to pass on adaptive traits. Resistance to a different species of bacteria, so being immune to antibiotics, for example, could provide a big boost for bacterium and fitness for that bacterium. So DNA analysis shows sciences which genes have been passed from one species to the next. So think of symbiosis. Okay, so two organisms of different species that have a very close relationship that involve living with each other is called symbiosis. So in endosymbiosis, one organism usually moves inside the other organism's cell. So scientists have theorized that endosymbiosis may be the mechanism that allowed life to generate on Earth. So mitochondria, the cell's powerhouse, and chloroplast that captures the sun's energy and stores it as food are both known as organelles of a cell. Such as a bacterium cell contains its own DNA and ribosomes, so do mitochondria and chloroplasts. Bacteria, mitochondria, and chloroplasts are also similar in size, so over millions of years of evolution, one type of bacteria became mitochondria and another type of bacteria became chloroplast. At first, many scientists rejected the idea that mitochondria and chloroplasts have evolved from bacteria, but finally advances in technology led to DNA sequencing that gave evidence supporting the hypothesis. So before all this, that organelles were seen under a microscope and the DNA and stuff, so scientists have been hypothesizing that chloroplasts and mitochondria were first bacteria that eventually evolved into becoming these organelles essential for cell survival that engulfed another cell and thus became the first true cell that could power, divide, and reproduce. We're going to move on to these other standards. Make sure you're also taking notes from CPUMs. They're very, very good with the lessons, and they also do have activities that you could follow. So remember, these are all review since you have talked about this before in 7th grade from your previous science class. So Gregor Mendel's ideas about inheritance and probability can be applied to all living things. Mendel determined that traits are inherited using pieces of information that he called factors and we call genes. So he observed and experimented with genes in pea plants, for example. He discovered how genes, such as those in ducks, were transferred from parents to offspring and how they made certain traits appear. However, Mendel did not know what genes actually look like. Remember, he was looking at these things long time ago. So remember, it was like the first microscope was being made, things like that. So you couldn't really, they were not very good. You could see cells, but you couldn't really go more in depth than that. Chromosomes are a lot smaller than the actual cell. Remember, the chromosomes and genes are going to be inside of our nucleus, so they were not really able to see inside of the nucleus just yet. Today, scientists know that gene segments of codes that appear in a structure called chromosomes. So these thread-like structures within a cell's nucleus contains the DNA that is passed from one generation to the next. These thread-like strands of genetic material have condensed and wrapped themselves around special proteins. This provides support for the chromosome structure. Chromosomes are also made at the beginning of the cell cycle. These series of events in which a cell grows, prepares for division, and divides to form daughter cells. During this time, the chromosomes get its characteristic X shape, just like we see here in this picture. Remember, the cell cycle is going to be this series of events that make up or are able to produce all of these new chromosomes. So every cell in your body, other than sex cells, has the same number of chromosomes. So in humans, the number is 46. Other organisms have different number of chromosomes. And there is a great variety. For example, mallard ducks have these 80 chromosomes. All sexually reproducing organisms form these sex cells, which have half the number of chromosomes that body cells have. So no matter what organism you have, you're still going to have these sex cells are always going to contain half of those. So for example, if you have an organism, let's say, I don't know, just whatever organism, let's say it has 200 chromosomes, so their sex cells are going to be containing not 100, those 100 set. Okay, so it's always going to be half. So every living thing needs to be able to have these instructions to survive. Without these instructions, living things would not be able to grow and function. These instructions are located on genes, which genes are located on chromosomes. So in humans, we have that 20 to 25,000 genes that are found in the 46 chromosomes. So chromosomes are different sizes. Larger chromosomes contain more genes than smaller chromosomes, and each gene contains instructions for coding a particular trait. There are hundreds to thousands of genes coding traits on any given chromosomes. From many organisms, these chromosomes come in sets. So all of these chromosomes that we see here in this picture, for example, scientists usually match them up. So for example, we have these 23 chromosomes as we see here. The 23rd would be considered as our sex chromosomes. So number 1, for example, would be matched up to what they are closely similar to. We see that 2 has this weird structure, but they're very alike. So pretty much you're trying to look at all of these chromosomes and match them up to your 46. So remember, one of them, or one side of it, is going to be coming from our parent or from our mom. The other side is going to be coming from our dad. We're just matching them up to see all of these pairs. Also with these pairs, during fertilization, you receive 23 chromosomes from your father and 23 chromosomes from your mother. So these chromosomes come in pairs called homologous chromosomes. They contain the same genes. 2 alleles, one from the mother and one from the father, represent each trait. However, the alleles for these genes may or may not be the same. Some of the alleles for how the gene is expressed may be dominant or recessive. So the offspring that receives these chromosomes inherited 2 different forms of a gene. So an allele, for example, A from one parent and an allele B from the other. So the individual can be heterozygous for that gene trait. Remember, heterozygous means that one of them is going to be dominant while the other one has recessive traits. That's going to give us that heterozygous figure. Also, we can have that homozygous. So homozygous means the same allele. So for example, you can have 2 dominant traits and be homozygous, or you could have 2 recessive traits that are going to also be homozygous. So that's also very important. Pedigree is so alleles can sometimes recombine to produce traits that are not favorable, such as a genetic disease, for example. So the geneticists study how these traits are inherited in order to trace their genetic origin and predict how they may be passed down to future generations. So a pedigree, what we have here is that picture. It's going to be a tool that geneticists use to map out the inheritance of traits. You saw one example of a pedigree in the case study. So the diagram shows the presence or absence of a trait according to the relationship within a family across several generations. It's kind of like a family tree, just for our genes. So we can see that we have our first generation, second, and third. Most of the time it doesn't really say, it doesn't have those. It's usually in Roman numerals. So most pedigrees show which family members express a particular trait, which is represented by the shaded figures, as well as individuals who carry the trait but do not express it. So those are the half shaded. In this scenario, and the picture that we have here, none of them are half shaded, but if they were, that means that they are carrying that trait in particular. In a pedigree, males are represented with squares and females are represented with circles. So one horizontal line connects the parent couple and another line leads down to the parents to their children. So for example, we have this first generation. So we have here these parents and these parents. So we have, this is the mom, this is the dad. Remember, the dad is the one that's carrying this trait. So remember, because it's shaded, so it's going to be expressed. So let's say that the gene is, let's say black hair, just to put it out there. Let's say that's black hair. And let's say that the mom has blonde hair. So we see that they had 4 kids. So in these 4 kids, for example, we have 3 boys and 1 girl. So we have here that this girl and this 1 boy have this trait, and it is they have black hair. Well, these 2 boys have blonde hair just like mom. So that's how you would read these. So forming these sex cells has a process called meiosis. It's pretty similar to mitosis itself. Just remember that meiosis makes these half of the chromosomes. So also meiosis occurs in a process that pretty much repeats itself, and it also does have this crossing over, which mitosis does not have. Mitosis creates identical cells. Meiosis makes cells that are not necessarily the same. So if an organism that is reproduced sexually, a body cell has twice as many chromosomes as a sex cell. So why is this important? Well, it is through the sex cells that parents can pass their genes onto their offspring. So when the sperm and the egg fuse, they form a zygote or a fertilized egg. The zygote gets 2 sets of chromosomes, 1 set from the sperm and 1 set from the egg. So human eggs, for example, contain the 23 total chromosomes in a set, and the sperm contain 23 total chromosomes in that set. So each of the body cells contain 1 set of chromosomes from your mother and another set from your father, for a total of 46 chromosomes. Sex cells, so sperms and eggs, are formed through a process called meiosis, during which the number of chromosomes is reduced by half. It is through meiosis that homologous structures separate into 2 different cells. This creates new cells with half as many chromosomes as the parent cells. Remember, the homologous chromosomes have 1 chromosome from each parent. While the 2 chromosomes share the same sequence of genes, they may have different alleles. Before the chromosomes separate and moves into the separate cells, they undergo a process called crossing over, just like that figure shows. A small segment of 1 chromosome exchanges places with the corresponding segment of the other chromosome. So by exchanging this genetic material, or genetic information, the new cell that forms will have a slightly different combination of those genes. This allows for minor variations of the traits to form, and also means that there is a higher likelihood for that offspring with desirable traits will form within a larger population. So remember, it's going to this crossing over. So you see that these 2 parts are the same. So they're going to exchange that genetic material. So we see that this white portion on the right-hand side went to the left, and the black portion went to the right. So they're exchanging the same types of alleles. They just do this in order to get an increase in variation. So that's the point. It was very important for meiosis, because none of us look exactly the same because of this crossing over that occurs only in meiosis. Mitosis makes exact cells. Just make sure you understand that. We're going to go over mitosis very, very quickly, because it is it's very simple. So you should already know the main parts of mitosis, which is prophase, metaphase, anaphase, telophase, until it gets to cytokinesis. So the prophase stage, we have the chromosomes that are condensing. So normally they're all over the place, and now they are condensing into these chromosomes, these egg shapes that we normally see. The centromeres starts to appear. So the centromeres are those small little kind of like rectangular shapes on either side of the cell that is going to be moving these chromosomes to their destinations after. We have metaphase, which are where our sister chromatids line up in the middle of these spindle fibers. So you're going to see all these X's lined up in the middle for metaphase. Anaphase, we're going to see the chromatids that are going to be starting to be pulled apart, that elongate the cell. So these chromatids are going to be pull to different parts of the cell, you're going to see these two distinct nuclei starting to move, and then finally we have this telophase where the nuclear envelope material surrounds each set of the chromosomes. So we're going to have this pretty much these the two nuclei forming on each set. And then at the end we have cytokinesis, which is actually going to be like our picture right up here, where we're going to have the splitting of the two cells. And then we're going to go back to meiosis and kind of compare them. So for right now, I'm actually going to pause here. So please make sure that you're taking very good notes on this. Make sure you're also looking at CPOMs for these standards. Thank you.